Cardiovascular disease is a leading cause of death in the United States and is mainly accounted for by myocardial infarction, stroke, and heart failure. Loss of energy homeostasis is a major contributor to cardiovascular disease. Cellular glucose transporters are linked to energy homeostasis. By facilitating the absorption and uptake of glucose, they regulate blood glucose levels. Two major families of cell glucose transporters exist: the facilitated diffusion glucose transporter, or GLUT family, and the sodium glucose cotransporter, or SGLT family. GLUTs are found in tissues throughout the body and facilitate passive diffusion of glucose down its concentration gradient. Expression of some GLUTs, such as GLUT4, are insulin sensitive. Within the SGLT family, SGLT1 and SGLT2 are responsible for glucose transport across membranes. These proteins couple glucose transport with the movement of sodium down its concentration gradient. Found primarily in the kidneys, SGLT2 is located in the luminal membrane of the proximal tubule, where it accounts for the majority of glucose reabsorption from filtrate. Located downstream of SGLT2 in the proximal tubule, SGLT1 similarly reabsorbs the remaining filtered glucose under normal physiological conditions. SGLT1 is also expressed in the intestine, heart, brain, and vascular endothelial cells. Less is known about the physiology of SGLT1 in the heart, brain, and vascular endothelium than in the intestine, where SGLT1 is responsible for the majority of dietary glucose uptake and is a regulator of postprandial plasma glucose. Dual inhibition of SGLT1 and SGLT2 is associated with reduced sodium and glucose reabsorption and increased urinary glucose excretion. Dual inhibition of SGLT1 and SGLT2 is currently under investigation for treatment in high-risk patients.